Good evening, everyone. Just make sure that everything is perfect from my side as well as from yours. Okay. Just tell me everything is perfect or not. Good. Okay. Okay. Let's wait for 30 seconds and we'll start. And do you have any doubts with respect to yesterday's class? And if you have any, you can ask me now. Do you have any doubts with respect to the cell division and cell cycle? You can ask me now. In today's class, we are going to see about this DNA replication, repair, and recombination. Okay. Okay, we'll start. What is DNA replication? And what is the major enzyme involved in the process of the DNA replication? Okay, first tell me whether I have to explain the concept first or we'll solve the MCQ first, and after solving all the MCQ, will I explain the concept, or shall I explain the concept first and after that we are going to solve the MCQ? Which one is okay for you? Concept first or MCQ first? Anyhow, I'm going to discuss the both, but you have to decide whether which has to be done first. Whether it will be the concept or will be the MCQs. Good, okay. I hope everybody is okay with that. Concept will be first. So, what is DNA replication? What DNA replication means? DNA dependent DNA synthesis. Okay, this is what a DNA replication. And what will be the central dogma? What will be the fundamental process is DNA has to be converted into RNA and RNA has to be converted into the protein. Okay, and DNA is converted into DNA. It is called as the replication. It is being replicated, and after that DNA has to be converted into the RNA. Okay, and this process is said to be as the transcription. DNA is transcribed into the RNA. This is called as the transcription, and RNA has to be translated to protein. This particular process is called as the translation. And one more important thing you have to remember is in prokaryotes and eukaryotes, this DNA replication happens in where? Where will be the exact location of this particular DNA replication is going to happen? In prokaryotes, this DNA replication happens in the cytosol. In eukaryote, this DNA replication happens in the nucleus. Okay. In prokaryotes, replication, transcription, translation, and post-translational modification, everything will be happened in the cytoplasm only. In eukaryotes, replication and transcription, post-transcription, and all the post-translational modification, including the RNA editing, everything will be happen inside the nucleus, and translation will be happen in the cytoplasm. Okay, this is what a basic phenomena. And okay, and what will be the major enzyme involved in the DNA duplication or the DNA replication? Means a DNA polymerase enzyme. Okay. Now, generally DNA is made up of, we know that sugar, it is made up of a nucleotide. Nucleotide is always present individually. Later it has to be combined with the three individual, particularly the sugar, phosphate and bases. This whenever the three substances, sugar plus phosphate plus bases, these three things combine together to form a nucleotide. Okay, and if the phosphate group is absent and the sugar and places has been combined, then we are going to call them as the nucleoside. Okay, and what is the basic difference between this RNA and DNA? DNA is said to be as the deoxyribonucleic acid, absence of oxygen in the second carbon. Okay, and in case of RNA, the oxygen is present. So we are calling them as the ribonucleic acid. And what will be the model? It is going to explain that what will be the two parent strands always associated with the daughter strands. This particular model will be the semi-conservative model. And who proposed this semi-conservative model means Matthew and Franklin Stahl. Meselson and Stahl, we can simply call them like that. Okay, and always replication will be start at the initiation point, and that is called as the origin of replication. Okay, and what will be the direction of a new strand synthesis? Replication will be started from which direction? Just tell me the answer, whether it will be in the 5' to 3' direction or in the 3' to 5' direction. In which direction the DNA will be replicated? 5' to 3' direction. And in what direction the proofreading will occur? Proofreading, or we can say the exonuclease activity. In which direction proofreading and exonuclease will activity will happen means 3' to 5' direction. Okay, and whether this replication will be occurs in unidirectional or bidirectional? Unidirectional or bidirectional? Bidirectional. And in prokaryotes, whether it is unidirectional or bidirectional? In prokaryotes, whether it is unidirectional or bidirectional? In both prokaryotes and eukaryotes, the DNA replication will happen in the bidirectional. Okay, it is always in bidirectional. And semi-continuous replication. Semi-continuous replication means you should know that always the daughter strand will be synthesized in the 5' to 3' direction. Okay, see. Sometimes the one of their strand will be the leading strand, okay, and other strand will be their lagging strand. Okay, whatever it is, whether it will be the leading strand or lagging strand or the Ogasaki fragments, always the daughter strand synthesis will be occur only in the 3' direction to the 5' direction. And in which strand, that is whether it is in a leading strand or whether it is in a lagging strand, which needs a more primer, which needs more primer. Which needs a more primer? Lagging strand. Lagging strand is the one, it needs a more primer because it is discontinuous. As it is discontinuous, always the DNA polymerase does not initiate a reaction, which means what? It can't initiate in adding the nucleotide. Somehow has to help that enzyme. So that DNA, that primase enzyme will add some of the nucleotide, 10 nucleotides in the first, and after that DNA polymerase will associate with it. Okay, as this lagging strand is discontinuous, for every strand DNA polymerase can't initiate a reaction. For that, it needs a different many primer to initiate a reaction. After that, DNA polymerase will continue to add the nucleotides. Okay, this is what we are calling them as the semi-discontinuous replication. And the second, last one is synthesis of RNA primers. This RNA primers will be synthesized by which enzyme? The name of an enzyme will be the primase enzyme. Okay, this primase enzyme, this is going to synthesize a short RNA sequences, which is called as the primers, approximately the 16 nucleotides, which is called as the in vitro. In 16 nucleotides will be added by this primase enzyme in the in vitro. Okay, but in our body, it will add approximately plus or minus 12 nucleotides. Okay. In a leading strand, there will be only one primer, but in the lagging strand, it needs multiple primers. Okay. Now, this is what about a DNA replication, a basic phenomena. First one is semi-conservative mechanism. As in a semi-conservative mechanism, we should know that all the daughter strand will have there some of the nucleotides, some of the strand from the parent strand. Okay, it has been conservative from their parents. So we are calling them as the semi-conservative, and they have been proved by this and N15 medium and the N14 medium. I hope you all know the experiment. And always its second point will be the ORI region, origin of replication region. When it comes to the prokaryotes, it has only one origin of replication site. And in case of eukaryotes, it has a multiple origins of replication. Okay, in eukaryotes, we are going to call that particular initiation region called as the replicator. Approximately, we are going to have 150 replicator. And in case of yeast, we are going to call them as the ARS region, that is autonomously replicating sequence. Okay, and the third one is always you should remember the direction of new strand synthesis. It will be 5' to 3' direction. Okay, and proofreading or the exonuclease activity will always happen in the 3' direction to the 5' direction. Okay, and the fourth important point will be the in whether it will be the prokaryote or it will be the eukaryote, always the replication happens in bidirectional. And the fifth point will be the semi-continuous replication, because it will be forming one leading strand and another lagging strand. Lagging strand is called as the discontinuous, and leading strand will be the continuous. So half of the replication will be discontinuous. So we are calling them as the semi-discontinuous replication. And the sixth point will be the synthesis of RNA primase. Always the DNA polymerase can't initiate a reaction. Someone has to help so that the first few nucleotides will be added by the RNA primer, and after that this DNA polymerase will continue to adding the nucleotides. These are all the basic thing about the DNA replication. These are all the rules of replication. And what are all the enzymes involved in the DNA replication? The first enzyme will be the helicase enzyme. Always the helicase enzyme will be involved in removing the this is going to unwind the DNA, because DNA is the double helical structure. It has to separate first, and after the separation it will be like this: 3' to 5', 5' to 3'. Okay, before the separation it will be like this. So the helicase has to be open, unwind the DNA strand so that the origin of replication site is visible to the RNA primer, and after the DNA polymerase will come on at the nucleotide. In prokaryotes, this helicase is called as the DNA B. In the eukaryote, it is called as the MCM complex. Okay, the name will be different when it comes to the DNA eukaryote and the prokaryote region. Okay, and the second enzyme will be the SSB protein. 1 minute. Helicase enzyme, it is the one going to unwind the DNA strands, and SSB proteins. This SSB protein has a 3 function with it. Okay, when it comes to the DNA replication, this SSB binding protein have 3 functions. One is it prevents re-annealing. Even after the helicase, it unwind the DNA strand into a linear strand. It can easily wind immediately. It can associate with it immediately. To prevent that immediate re-annealing, this SSB protein plays a major role to keep the DNA unwind. Right, and the second thing will be the if the DNA after it has been separated or after it has become unwind, it is easy to degenerate by some of the enzymes. So that degeneration by some of the enzymes to this DNA strand will also be protected by this single stranded binding protein. And the third one is prevent coiling. Okay, this DNA linear strand, it undergoes a coiling, and that coiling will be prevented by this SSB protein. And after that, the most important protein will be the topoisomerase enzyme. Topoisomerase, or we can call them as the gyrase enzyme. Okay, and this is the one also involved in prevent the coiling. Okay, they are also one going to prevent the coiling. Okay, and it has a different types, topoisomerase 1 and topoisomerase 2. This topoisomerase 1 will always prevent the coiling in the, they are going to cut only the single stranded DNA. Okay, always it prevents the coiling by cutting the DNA strand, and that cut will be happen only in the single stranded DNA in case of topoisomerase 1. In case of 2, it is going to cleave a double stranded DNA. Okay, and after that, a primase enzyme. The primase enzyme is also called as the DNA G in prokaryote. Here, this primase will be or this is the one going to synthesize the RNA primer. Okay, only after this RNA primer, the DNA synthesis will be continued by the DNA polymerase enzyme. Okay, and after that, the most important enzyme will be the DNA polymerase enzyme. In DNA polymerase, we have 3 different types majorly. Now they have been invented more, but majorly DNA polymerase 1, 2, and 3 will be there. And in prokaryotes, we are having DNA polymerase 1, 2, and 3. In eukaryotes, we are having 5 different types of DNA polymerase. One is DNA polymerase alpha, beta, gamma, epsilon, and delta. Okay, when it comes to the DNA polymerase 1, 2, and 3, 3 will be very effective. Why? Because it has a more processivity as well as the polymerization rate. Okay, what is processivity and what is polymerization rate? In case of polymerization rate, they are going to decide how many number of nucleotides has been added per minute. Per minute, how many nucleotides they are adding. That is what a polymerization rate, the speed of adding the nucleotides per minute. And in case of processivity, if this how fast they are adding, okay, how efficient they are adding, the ability or capacity. For example, the DNA polymerase per minute, they are going to add approximately 10 nucleotides. For an example, 10 nucleotides they are adding per minute. Okay, for how long they are going to sustain in the process for adding this 10 nucleotides per minute. Okay, how long they are sustaining, that is sustainable point will be the processivity, whether it is going to add the nucleotides for half an hour, whether it is going to add the nucleotides for 1 hour, like that. Okay, how long they are going to sustain, that is what a processivity. Okay, and which has a high processivity means obviously DNA polymerase 3 will be the will having the high processivity. And also you should know that in which enzyme has the capacity to do and proofreading even in the 5' to 3' direction. This is what an important point. See, anyhow, DNA polymerase 1, 2, 3. Everything will be undergoing in everything will be involved in adding the nucleotide. So everybody involved in the polymerization, and always the nucleotides will be adding from the 5' to 3' direction. This is common to all the enzymes. And if the proofreading, this proofreading always happens in the 3' to 5' direction. But in case of this polymerase 1, they have an extra characteristic features that it can also proofread in the direction of 5' to 3'. So this can be asked in the question. And also DNA polymerase 1 have the capacity to repair the DNA. This is also an unique point when it comes to the DNA polymerase 1. Okay, and all this DNA polymerase 2 and 3, they are not having any DNA repair mechanism. They are not having such capacity. Okay, this is what an important functions of this enzymes. Okay, that's all about a basic information about a DNA, and everything will be explained here. Okay, now come back to the question. Which enzyme is responsible for unwinding the DNA double helix during replication? helicase enzyme. Let me explain you a little more extra about a helicase enzyme because there are different types of helicase enzyme. In prokaryotes, we are calling them as a DNA B, but see how they are going to unwind the DNA. This is what an important mechanism. They are going to melt the hydrogen bond. Okay, which bond they are going to melt? They are going to melt the hydrogen bond. Okay, and always this helicase is the one going to associate, they are going to identify the origin of a replication region. Okay, and they are having DNA A, DNA B and DNA C. They are the types of the helicase enzyme. Okay, A is the one going to identify the ORIC region, origin of replication region, and after that B and C is associated with this A, and wholly we are going to call them as the MCN complex in case of eukaryotes. Okay, and this helicase has the capacity to unwind the DNA, and the speed will be 1000 base pairs per second. 1000 base pairs per second. Okay, and always this helicase will move in a direction, and that direction is called as the replication fork. Okay, and its same direction the strand produces, it is called as the leading strand. Okay, the helicase moves towards me. This is called as we are going to call them as a replication fork. Always the leading strand will be produced along the direction of replication fork. But in case of lagging strand, they are going to produce a daughter strand against the replication fork. Okay, this is what an important mechanism. And one more point is DNA helicase will be the ATP dependent enzyme. It needs an energy to unwind the DNA, and that energy will be produced by the phosphate bond, that is ATP. Okay, clear? I hope everybody will be clear under this. And let me explain the order how the enzyme will work. Okay, first initially the helicase will come. Okay, helicase will come and unwind the DNA, and after that SSB binding protein will come and prevent the immediate joining of the DNA strand. And when it do such thing, it undergoes a coiling at the same time. The DNA undergo a coiling, and that will be prevented by the topoisomerase enzyme. In prokaryote, we are going to call them as the gyrase enzyme. Okay, and after that RNA primer will come and associate with it. Okay, and after that DNA polymerase will come and add the further nucleotides. And finally, the DNA polymerase is the one. The DNA polymerase 1, okay, DNA polymerase is the one is going to remove the primer. The primer has to remove. Okay, and this primer has to be going to remove by the DNA polymerase 1. Okay, DNA polymerase 1, and after that that nick will be sealed by the DNA ligase enzyme. See, initially helicase will come and unwind the DNA, and after that this SSB binding protein will prevent the immediate joining of the DNA strand, and after that the supercoiling will be removed by the topoisomerase enzyme, and the RNA primer will be adding, and the DNA polymerase 3 is going to add the nucleotides, and after that this DNA primer has to be removed, and that has been done by the DNA polymerase 1 enzyme, and after that that nick will be sealed by the nick will be sealed by the DNA ligase enzyme. Okay, this is what the order how the enzyme will work in the replication process. Which direction does DNA polymerase add new nucleotides during the replication? That is 5' to 3' direction. Always, whether it will be the leading strand or it will be the lagging strand, always the direction will be the daughter strand will be synthesized in the 5' to 3' direction. What is the role of primase in DNA replication? What is the role of primase in DNA replication? We know that that will be the RNA primer. They are the one going to synthesize the RNA primer. Okay, in our body, approximately plus or minus 12 nucleotides. In the in vivo in vitro condition, that is in the laboratory, we can produce up to 60 nucleotides. Okay, which is the one going to join the Okazaki fragments? That is the lagging strand. See, it is going to synthesis like this. This continues. How these strands will be joined? How these Okazaki fragments will joined? Joined by which enzyme? Ligase enzyme. And how it is going to join? How it is going to join? What is the name of the bond? This ligase can join the fragments by which bond? Ligase is going to seal the neck by using which bond? Is it so? Is it a hydrogen bond? Everybody, Basha, just answer for this question. Very good. Phosphodiester bond. Okay, one nucleotide will be associated with the other nucleotide through this phosphodiester bond. Okay, see, always we can say bases. Okay, bases, adenine, guanine, cytosine, thymine. Okay, this is going to bond like this. And which is what is the name of the bond? Here adenine is always paired with diamond using the hydrogen bond. Okay, and double hydrogen bond, double bond will be happening here. And cytosine and guanine will always bond with the triple bond. And what will be the name of this bond? In using which bond these bases are getting connected? Hydrogen bond. Here comes the hydrogen bond. Let's see, bases, sugar, and phosphate group. Okay, a glycosidic bond will be involved here, hydrogen bond will be involved here, and the phosphodiester bond will also involving here. Okay, this is comprising the one nucleotide, and another nucleotide will be like this, and this two nucleotide will be adding using the phosphodiester bond. Here one nucleotide will be there, here one nucleotide. These fragments will be joined by the DNA ligase enzyme using the phosphodiester bond. Which of the following is not a component of DNA replication fork? Not a part of DNA replication fork. Helicase, DNA ligase, single strand binding protein, RNA polymerase. Okay. RNA polymerase is involved in the transcription process. We know what is the function of helicase, DNA ligase, single strand binding protein. RNA primer will be involved here, but not the RNA primase. Okay, this is what somewhat related in the transcription process. And what are all the components involved in the replication fork? DNA replication fork means helicase, DNA polymerase, primase, DNA ligase, single strand binding protein, and topoisomerase. These are all comprisingly called as the DNA replication fork. And always towards the direction of replication fork. All these enzymes comprisingly called as the replication fork, and towards the direction of replication fork, the daughter strand will be synthesized, and that particular strand will be calling them as the leading strand. Okay, and against the direction of replication fork will be the lagging strand. What is the purpose of DNA ligase in DNA replication? Who is going to unwind the DNA double helix? Synthesis of the unwinding the DNA helix will be done by the DNA helix. Synthesis of RNA primers will be done by the primase enzyme. Joining the DNA Okazaki fragments will be done by the DNA ligase enzyme. Proofreading will be happened by the DNA polymerase. All the polymerase will going to proofread. But DNA polymerase 1 has the capacity to do a proofreading in both directions, 5' to 3' as well as in the 3' to 5' direction. Okay. DNA polymerase 1 has many functions. Kindly have a look at that. Which type of DNA repair mechanism? Before we go into that, I'm going to explain what is meant by DNA repair mechanism. Okay, how the DNA is going to damage. How many types in DNA repair mechanism? We have a different types of repair mechanism will be there. We have a depurination or the appurination or the A basic site. Okay. See, first DNA repair mechanism will be the depurination. Depurination, which means what? Loss of a purine basis. See, kindly make a note. What are all the points I'm going to explain? That is not in the PPT. Let me explain. Kindly make a note of it. Okay. And the first type of DNA damage will be the depurination or the appurination, which means what? Loss of the purine basis. What are the purine we are having in the nitrogenous bases? Generally, we are having the 4 nitrogenous bases. Is it not? Adenine, thymine, cytosine, and guanine. Okay, in these 4, what we are considering as the purines. Adenine, thymine, cytosine, and guanine. Purines are what? Adenine and guanine are considered as a purine. So, if we are losing the purine, then we are calling them as the depurination or appurination. Okay. And in depurination, it is the DNA damage, and this damage has to be repaired. Okay, and that repair mechanism will be the base excision repair. Okay, base excision repair pathway. And the second one will be the deamination. Deamination means a removal of amine group completely. Okay, that amine group will be removed in that DNA. And on this removal, one base can be completely converted into an unnatural base. Okay, see, one base. Base means what? For example, a cytosine. Cytosine is converted into an uracil. What is the purpose of having an uracil in DNA? And this is the base which has to be present in the RNA. But because of the DNA damage, we are having the deamination. If the amino group has been removed, the cytosine has to be converted into the unnatural bases. For example, in case of cytosine, it has to be converted into the uracil. In case of methyl cytosine, it is going to convert to the thymine. In case of an adenine, it will be converted into hypoxanthine. In case of guanine, it is going to convert into xanthine. So, the bases are converting into the unnatural bases. This is what somewhat dealing with the deamination. Okay. Now, the third type of DNA damage will be the oxidation. What is it? Oxidation. Here, this DNA will be treating with the oxidizing agent. During the treating, the entire base sequence has been changed completely because of the oxidation process. And this oxidation cause a most severe damage when compared to the deamination and depurination. Okay. Here, when the guanine undergoes an oxidative stress, okay, it is going to convert into 8-oxoguanine. This is the most dangerous when compared to the above two. And the fourth one will be the alkylation or methylating. Alkylation or methylating. This when DNA is treating with the different types of alkylating agent. Okay, and if we are going to convert if we're going to treat this DNA with an alkylating agent, mean they are also going to convert into an unnatural basis. For example, methyl nitrosourea and nitrosoamines and some of the drugs like temozolomide. These are all the name of the alkylating agent. For example, the cytosine. Again, the cytosine, if it is treating with an ammonium hydroxide, they are going to convert them as the hydroxyl aminocytosine. And always the cytosine has to be paired with the guanine. But when it is treated with some of the alkylating agent, it is converted into the hydroxyl aminocytosine. And this hydroxyl aminocytosine always pair with the adenine. Okay, like in the same way, if the guanine is treating with the methyl methyl methane sulfonate, at its sixth position, this MMS add the methyl group. So this guanine will be converted into the O6 methyl guanine. And always this guanine has to be paired with the cytosine. When it is treated with the methyl methane sulfonate, it is converted into the O6 methyl guanine. Now this O6 methyl guanine has to be paired with the thymine. Okay, so the pairing mechanism has been changed here. This is what a fourth DNA damage. And the fifth one will be the dimer formation in bases. Formation of dimer. For example, a thymine dimer. When it is when the DNA is exposed to the UV rays, okay, this is the one going to cause a disease like melanoma, skin cancer. Okay, see for example, thymine. If two thymine are going to add, and in the presence of UV, they are going to form a thymine dimer. Okay, it is also called as the CPD dimer, cyclobutane photodimer. Okay, it is looks because its structure will be looks like a cyclobutane. Okay, so thymine dimer will always cause the skin cancer. This is what a fifth type of DNA damage. And the sixth type will be the mutation caused by the base analogs. Base analogs means the normal base, we are having adenine, thymine, guanine, and cytosine. We are having the four bases. And the base analog means structurally they are going to similar with the normal bases. For example, 5-bromouracil, and you have to remember that we are having adenine, thymine, and guanine and cytosine. Say for an example, the base analogs means structurally they are similar to any of these four nitrogen bases, but they are not functionally same. Okay, structurally same and functionally not same. This is what we are calling them as the base analogs. And for example, 5-bromouracil. 5-bromouracil, they are the one behaving like a thymine. They are looks like a thymine in the normal state, and it's paired with the adenine in the normal state. Okay, but it behaves like a cytosine in the rare state. Okay, and pair with the normal state. It's just a base analog of adenine. Okay, you should remember that this is the base analog, structurally same. And if somewhat this adenine has been converted into 5-bromouracil, its function is going to different, and again it also considers a DNA damage. And the seventh type will be the mutation caused by the intercalating agent. Okay, intercalating agent means on its own introduced in DNA, they are going to randomly choose the base pairs. For example, ethidium bromide, acridin orange. See, these are all the most different examples. So you can easily choose the intercalating agents if they have been asked in the examination. Ethidium bromide, acridin orange, actinomycin D. Okay, these intercalating agents going to damage the double helical structure of the DNA. Okay, so if it is caused the DNA at the double stranded structure itself, they can't undergo a DNA replication. They can't undergo a translation, transcription, or not even a repair. Okay, they're not going to happen if the mutation caused by the intercalating agent. These are all the types of DNA damage. And also, this intercalating agent, they are causing a deletion or insertion. Some of the bases has to be deleted or some has to be inserted. Okay, this is finally we can call them as the frame shift mutation. This frame shift mutation is called caused by the intercalating agent. Now comes the DNA repair mechanism. Now what are all the things we have seen before was DNA damage. Now we are going to see how we are going to repair that particular DNA damage. There are different types of DNA repair mechanism will be there, and the first type will be the mismatch repair. Mismatch repair, and this will be the, see, sometimes the DNA polymerase, we are calling this DNA polymerase as the most accurate enzyme. Okay, they are not going to do any mistakes, and sometimes they are going to done a mistakes 1 into 10 power 6 nucleotides. Okay, they are considered as the most accurate enzyme. Okay, and if it do any mistakes, the entire DNA sequence will be will have an impact. Okay, this can be repaired by the principle of methylation. Okay, this methylation, for every 1000 nucleotides, at least a few palindrome sequence will be present. Okay, see, palindrome sequence in the DNA strand will be seen very oftenly. Okay, we should not think that this palindrome sequence is very rare in the DNA, but instead that we have seen a lots and lots of palindrome sequence in the DNA. That palindrome sequence got methylated. Now they have been considered that now the damage is in the daughter strand. If you want to repair the daughter strand, all the mismatch repair mechanism enzyme have to differentiate whether the parent, whether it will be the parent strand or the daughter strand. So for that differentiation, this daughter strand has to be methylated. If the parent strand has a palindrome sequence, the daughter strand also has a palindrome sequence. So if the parent strand has methylated and the daughter strand will not be methylated, we are calling them as the hemi-methylated DNA. After some time, certain time, it will be methylated by the with the help of an enzyme called the dimethyl transferase enzyme, because they are the enzyme is going to add the methyl group to the palindrome sequence of the daughter DNA. By seeing that, we can easily come to other repair when the DNA is hemi-methylated. Okay. Okay. Now, there are many enzymes involved in the mismatch repair, and this mismatch repair is done by the how they are going to differentiate this daughter strand or the parent strand is by the methylation process. Okay, and second one will be the base excision repair mechanism. In case of base excision repair mechanism, this they are going to remove the damaged bases and replace them with the correct ones. And this base excision repair, this repair will be repaired if the DNA is damaged by the process of deamination. Okay, if the deamination will be happened and the DNA has been damaged, means this base excision repair is going to come. This is the mechanism is used to correct the damage done by the deamination process. And what will be the, see, it is this DNA base excision repair mechanism will be initiated by the DNA glycolase enzyme. Okay, to break the unnatural bases, and the N glycosidic bond has to be broken to remove the unnatural bases. Okay, this, and also you should know that this DNA glycolases is an enzyme. It is very specific. Okay, it is very specific to which at what kind of DNA it has to be added. Okay, this is the one used to remove the unnatural bases. Okay, and if uracil has to be added, the uracil DNA glycolase enzyme will come into a picture. If xanthan is introduced, then xanthan DNA glycolase will be come into a picture. And this DNA glycolysis first cut these unwanted bases, and after cleaving the base, the phosphodiester bond is to be cleaved, and after that, the nucleotide has to be replaced, and there will be a gap called the apyrimidinic sites or the apurinic sites. Okay, and that gap will be sealed by the DNA ligase enzyme. Okay, now the second one, third one will be the nucleotide excision repair. And here, if the damage caused by the UV rays, Okay, if the damage caused by the UV rays, then the repair mechanism followed here is nucleotide excision repair. And same thing, the mismatch repair, they are the one correct the errors. If the error done by the DNA polymerase enzyme. If the error done by the DNA polymerase enzyme, then the mismatch repair will be come here. And if the damage caused by the UV rays, then the nucleotide excision repair will be happen. And if the damage caused by the deamination, if any unnatural bases will come, will be caused the any damage, then base excision repair will be followed by an enzyme, DNA glycolysis enzyme. Clear? And in case of nucleotide excision repair, XC nuclease enzyme come into a picture. XC nuclease enzyme will come into a picture. And in case of this mismatch repair means there are different enzymes, that is mute L1, mute L, mute S, they are all the DNA repair enzyme. Okay. Okay, there are different types of enzyme involved in the mismatch. They are called as the mute H, mute L, mute S. These are all the enzyme involved in this type of repair mechanism. That's all about the repair. And what is meant by direct repair? Direct repair means whatever the damage caused by the UV light, that can be repaired by using a visible light. Okay, direct repair means whatever the repair done by the UV light, this damage can be repaired by using a visible light. So the direct repair mechanism is also called as the photoreactivation. Okay, so this diamond dimer can also be repaired by this nucleotide excision repair as well as the direct repair, but the definition was different. Okay. Direct repair is somewhat using the visible light, we are going to repair the damage caused by the UV light. But in case of nucleotide excision repair, the XC nuclease enzyme will come into a picture, right? Now, SOS repair, and that is a different phenomena. We don't need that. Just know that is also a repair mechanism. Now, come to the question, and which type of DNA repair mechanism corrects the DNA damage caused by the exposure to the UV light? Nucleotide excision repair. Okay, what is the end product we are going to get at the result of this UV light? Thiamine dimer. Okay. Which enzyme is responsible for recognizing and removing the mismatched base pairs during the DNA repair? Recognizing and removing the mismatched base pairs during the DNA repair. Is it so? Is it so? Mismatch repair protein complex. Okay, they are the protein complex. They are going to detect and remove the mismatched base pairs that occur during the DNA replication. Okay, and here the mute S, mute L, and mute H will be happen. Okay, very good. And which type of DNA polymerase involved in the DNA repair mechanism? Which type of DNA polymerase enzyme involved in the DNA repair? DNA polymerase 1, very good. What is the role of DNA polymerase in base excision repair? It recognizes and removes the damaged bases. It exercises the segment of damaged DNA. It synthesizes new DNA to replace the damaged segment. It seals the neck in sugar phosphate backbone. Think carefully and answer the question. Okay, it is very easy. What is the function of the DNA polymerase? Yeah, obviously, it is going to synthesize the new DNA. Why? Because the function of this DNA polymerase is to add the nucleotide. So, obviously, it has to synthesize a new DNA to replace the damaged segment, right? Now, which DNA repair mechanism is primarily responsible for repairing double strand breaks? We know base excision, we know nucleotide excision, we know mismatch repair. Okay, by this, even though you didn't know about the homologous recombination repair, you can easily answer this question by eliminating an option. See, double stranded. We didn't see any this type of breaks in these type of repair mechanism. So the answer will be the homologous recombination repair. It is a primary mechanism for repairing the double-stranded break. An important statement. See, and this repair will be done particularly during the ES phase, G2 phase, because we are having a different checkpoints at these stages. Okay, during the cell cycle. It uses the undamaged sister chromatid or the homologous chromosome. That why it has a name homologous recombination repair. Hope you are clear with this. Okay. What is the function of the DNA ligase in DNA repair? Quick, you should not think this much for this question. It is going to seal the neck in the sugar phosphate bond. And which technique is used to amplify the specific DNA sequence in vitro? Which technique is used to amplify a specific DNA sequence in vitro? Okay, polymerase chain reaction. We know that this is a technique to amplify the specific DNA. And what are all the steps involved in the PCR? Denaturation, annealing, and extension. And this will be a different topic. And what is the function of a restriction enzyme in recombinant DNA technology? What is the purpose of doing this recombinant DNA technology? We are going to clone a cell, okay, into a many copy as a result of this recombinant DNA technology. I hope you are familiar with this topic. I don't want to explain this. I'll simply send this PPT to you, okay, as this time is very short here. I couldn't explain. Always the restriction enzyme is involved to cleave. Okay. Which vector is commonly used for cloning of large DNA fragments? Quick. Large DNA fragments. Always see the donor DNA will be there and the recipient DNA will be there. The donor has to be enter into the recipient DNA and it can't enter directly, but instead of that we have to use this plasmid or the carrying vehicle or the vector to take this donor DNA, and the plasmid with the donor DNA we are going to insert in this recipient DNA. After that, the recipient will think that this DNA is our DNA and they are going to multiply the donor DNA within the recipient. Okay, as a result, they are going to give a different copies. But what the question asked here is with respect to the vector. Which vector is used to clone a large DNA fragment? And it obviously is the artificial chromosome. Okay, and what is the purpose of selectable marker in recombinant DNA technology? Okay, wait 1 minute, wait. I'll explain that topic first, and after that we'll discuss this question. Okay, there are different terminologies over here. Okay, I'll explain this topic tomorrow, and after that we'll discuss some of the questions. Hope the session might helpful, and if you want to ensure something, you can tell me now. And do you have any doubts with respect to this DNA replication? And I didn't explain the steps, that is initiation, elongation, and termination. In initiation, they are going to start the point at the origin of replication. Okay, where they are going to find the region origin of replication. They are going to there we can find the replication as a starting point. And in elongation, the DNA polymerase is going to add the nucleotides in the direction of 5' to 3' direction. Okay, and termination, we are going to terminate at the particular site. I'll send the PPT, don't worry. Anything else you want to know? PPT will be shared immediately after the class. Don't worry. Any clarifications? Okay, I have shared. Okay. Anything else? Okay, done. Shall we wind up the class? For tomorrow, what's our topic? This recombinant DNA technology, transgenic, everything will be, this line will be explained tomorrow. And also you have to read these two, protein synthesis and regulation of gene expression. I think LAC operon has been completed. Only lambda operon will be there. Okay, we'll finish all the remaining topics in the cell biology. And you have to revise all these topics. All right? All right? Okay. It is about time to wind the class. Okay, done. We'll meet you tomorrow the same time. Thank you. Thank you, everyone. And if you have any doubts, you can post me in the group.